What are nucleases? Nucleases are enzymes that can cleave, cut, the phosphodiester bonds between nucleotides in nucleic acids, DNA or RNA. They help in DNA/RNA processing, repair, replication, recombination, and degradation. Types of substrates: some act on DNA, some on RNA, and some on both. Types of nucleases. Nucleases are classified mainly into two categories. 1. Exonucleases. Cut nucleotides from the ends of DNA or RNA molecules. They remove nucleotides one at a time, either from the 5' or the 3' Examples: Exonuclease I removes nucleotides from 3' of single-stranded DNA. Exonuclease III removes nucleotides from 3' of double-stranded DNA. RNase II acts on RNA molecules. Properties of exonucleases. Sequential removal of nucleotides. Can be 5 rightward arrow 3 or 3 rightward arrow 5 acting. Important for proofreading during DNA replication and in DNA repair. 2. Endonucleases. Cut within the DNA/RNA strand, not from the ends. They cleave internal phosphodiester bonds at specific or random sites. Properties of endonucleases. Breaks DNA/RNA inside the chain. Can create blunt ends or sticky ends depending on the enzyme. Can be sequence specific, like restriction endonucleases, or non-specific, like Nuclease. Used in genetic engineering, restriction enzymes. Examples: restriction enzymes, EcoRI, Hind III, sequence-specific endonucleases used in recombinant DNA technology. Nuclease cuts DNA randomly into smaller fragments. RNase cleaves RNA internally. What are endonucleases? Endonucleases are nucleases that cut nucleic acid chains at internal positions, breaking the phosphodiester bonds within the molecule. Special feature: they do not require free ends to act, unlike exonucleases. Types of endonucleases. Non-specific endonucleases cut DNA/RNA anywhere, example Nuclease I, S1 nuclease. Restriction endonucleases, restriction enzymes, recognize specific DNA sequences, usually palindromic, and cut at defined sites. Type I, cut at random sites, away from recognition site. Type II, cut at recognition site, most widely used in molecular biology. Type III, cut near the recognition site. Applications of endonucleases. Gene cloning, restriction enzymes. DNA footprinting. Nucleic acid mapping. Genome editing, CRISPR-associated nucleases like Ca9 are specialized endonucleases.